Imaging One World. Oh. Welcome to today's Imaging One World talk. Now that now recording has started, so which reminds me that we've now put a lot of the talks on YouTube. So if anyone's wondering what TV series to watch next, then I can recommend Imaging One World on YouTube. It's good variety of movies there.
You're gonna have to watch the Squid Game.
Indeed. So today, anyone who's got any ideas for Imaging One World speakers, we'd be great to receive them. You can email any of the organisers, and we'll look forward to that. We're just planning from January to Easter for our next series of talks. So all ideas welcome. So today, I'd like to welcome Susan Cox, who is at the Randall Center for Cell and Molecular Biophysics, and she's known for a lot of kind of like software development and super resolution work that over many years has developed into some very interesting techniques that including Hawkman, which has just recently been published, which I'm sure she'll be telling us all about. So instead of me telling you about Susan's history, it's probably best if Susan just talks and tells us about it herself.
Thank you, Alex. It's a great pleasure to be here, and I want to thank the organisers for the invitation. I've really enjoyed the series so far, and I hope you all enjoy today as well. So without any ado, I'm going to just start sharing my screen. Can everybody see the shared screen?
Yes.
Yep.
Amazing. Thank you, Alex. Okay, so I want to take you on a journey today through some work that we've been doing, a little bit through the history of our ideas of what we did in localization microscopy at various times, and how you can use the various ideas that we've been developing to get from initial raw images to information, and how you can assemble that information into something where you've enhanced the resolution of the image and improved the accuracy of your answer, and maybe also to spend some time pondering what resolution is and the relationship between resolution and accuracy and biological structure and information. So I've spent a lot of time thinking about resolution over the past 10 years, and the longer I spend thinking about it, in many ways, the less I feel that I know. And in fact, after a while, I end up feeling like Mr. Spock on encountering weird alien species such as humans, and I wonder, what is this thing that we're calling resolution? What does it actually mean? Now, in a wide field microscope or a confocal microscope, this concept is pretty well defined. And in a wide field microscope, the Abbe limit, if that's how you choose to quantify resolution, is defined as the emission wavelength of the fluorophore that you're using divided by 2 times the numerical aperture. So this is really just about the optical properties of the system you're using. And that defines the size of the point spread function, which in turn defines how close 2 point spread functions can be to distinguish them. Now, it's worth noting that, of course, this is based on a subjective assessment of how close 2 things can be, and you can still tell them apart. If you had a really, really bright signal, and you could perform modeling, you could pick out 2 things that are much closer together than that. If you had very poor signal to noise, if you had only a few photons, things would have to be much farther apart for you to tell that they are actually 2 separate things. It very much depends on the quality of the information as well. But this is the traditional definition, and so we'll be using it. For SIM, we know at best, we get at best half this value. And for STED, you can make a definition related to the intensity of the depletion beam. And these are all fairly easy to interpret. But I'm going to be talking today about localization microscopy. And in localization microscopy, the resolution, to the extent we can define it, varies locally with the properties of fluorophores, the distribution of the fluorophores, and what knowledge we've built in when we've modeled the data. So there isn't a resolution for a localization microscopy image. There's a property of the image that varies across it. And this is going to be one of the issues we're going to be tackling today. So I want to enter a little bit more into this idea that knowledge is resolution, because I think it's a really useful way of viewing localization microscopy. So if we know that we're getting out of focus blur, then we can perform a process called deconvolution on our images. And this was in fact done all the way back in the 1980s. Here's an example by Agard and Sadat, where they took the images that are on the bottom row of a zebrafish embryo, and they then created a model of how out of focus light is propagated through the sample. They then removed the intensities that they calculated for the out of focus light, and what they got was the image that's on the top, which look a lot sharper. So great, you've got much sharper images. Is that enhanced resolution? Well, it's clearly enhanced ability to discriminate structure. Maybe what's been done more recently is, if you know the point spread function, then you can deconvolve. You can form a model of your point spread function, and you can use that to try and undo the process of the combination of information between the point spread function and the sample. And it's really important to note that you here are performing a model and you are building in assumptions. So here we've modeled the imaging process as consisting of a finite amount of data formed from a continuous object. This is some work from Carrington in 1995. And you can see you can get images that are very sharp. You can see peaks that are separated by about 110 nanometers, but you have built in information that you are assuming is correct, that you have a continuous object. That means you will not see if there are any breaks at or below the diffraction limit in your sample, which means in turn, you're not really adding any information. You're enhancing the sharpness of the image, but you're not able to extract any extra information. You're just making it a little bit easier to see what the structure is. Okay, so localization microscopy. In localization microscopy, how are we achieving an improvement in resolution? Well, we're separating fluorophore emission in time. That is, we're only having a few fluorophores emitting light in each frame, and we're then localizing each fluorophore. And localization microscopy exploits the fact that a feature of a known shape can be localized to a much higher precision than the resolution of the system. And our knowledge of the expected shape gives the resolution boost. So here I'm showing a point spread function. I'm showing on the right a cut through of the data with intensity on the y-axis, and the resolution of the system is determined by the width of that point spread function. And if we then perform a fit, then the accuracy with which we know the position of that molecule is limited by the mismatch between the red fit and the blue data. And you can see that mismatch is much, much smaller than the width of the peak. And this is where the enhancement comes from. And the improvement in resolution is determined by the number of photons you can collect from that molecule and the background level. So basically the signal to noise from that molecule. So this is great. We can find the position of individual molecules, but of course we were able to do this back in the 1990s. You could track individual single molecules. The challenge is you don't tend to want to view images of samples with just one or two molecules. You want to view an image with a whole load of molecules at once. And to do this, you need to induce blinking, and other people spend a long time determining how to induce blinking effectively. And here I'm just going to assume that this is a problem we are able to solve. There's a number of possible solutions. the most common nowadays still being to use organic dyes and a buffer, or something that's been developed more recently, DNA origami, where you use binding, unbinding. But there are many people who still use photo-switchable fluorescent proteins because you can then use them in live cells. So it's something where the technique very much depends on the experiment that you want to run. So, okay, we're able to get data where we have fairly well separated molecules in each frame, but it's really important to remember that this excitation process is stochastic. You don't have any direct control as to which fluorophores are lighting up. And that means that there's a good probability that at some point you're going to get fluorophores overlapping. You also have a trade-off between how many fluorophores on average you're exciting in each frame of the raw data and how many frames you have to take to get a good image. You could decrease the probability of getting overlapping fluorophores, but it would take a hugely long time to collect data because you'd have to go down to a very tiny number of fluorophores per frame. Or you could collect a lot, but you increase the probability of getting overlapping fluorophores. Now we started to think, how does this issue of activation density work mathematically in terms of optimizing your experiment? And we started to think about starting off from a very low density of activation and then gradually ramping the density up until it's very, very high as a thought experiment. So here on the left, I'm showing the raw data, and on the right, I'm filtering out the data after about 30 frames to let you see how many fluorophores are being localized to that density. So you can see it starts out, I'm just going to restart, pretty low. Then as I increase the activation density, the localized density gets higher. And then as I increase the activation density further, the localized density falls again. So it appears that there's an optimum activation density to get the best performance out of your localization microscopy system. But it's important to note that the optimal activation density will also depend on the structure. I've realized that I put another slide in here. I'm going to just diverge. It's important to know that the reason why you might care a lot about getting the activation density right isn't just that there's an optimal density for activation where you can do things fastest. It's also important because if you get it wrong and you have a very high activation density, you might well get artifacts. And there are, of course, many different sources of artifacts. And I'm just showing two examples here. One from Ritzley, where he has looked into fixation artifacts in a lot of detail, and one from looking at analysis artifacts. This is also an issue that's been explored in quite a lot of detail by Sauer and others over the years. But it's important to note that when you go to higher densities, you don't just get a degraded resolution image of your sample, you get something that looks different. And we're going to be coming back to that in a little bit. But let's come back to this idea of an optimum activation density. So we thought about how this would work for different types of structure. So I've got here examples of 0D samples, 1D samples, and 2D samples. This might be something like small clusters, microtubules, and focal adhesions. And then we just looked at activating each of them with the same number of fluorophores per unit area. And then we looked at how close the fluorophore activations were and classified them as either successful in yellow or unsuccessful in blue. And you can see that what we get is actually fewer successful fluorophore fits in the focal adhesion R1, even though we have many more activations. So this happens because we are limited not by the fluorophore we're imaging at the moment, but by the number of fluorophores that we need to image, but can't because they're too close. And by too close, I generally mean within about 450 nanometers for most of the single molecule fitting algorithms. And for multiple emitter fitters, you can get down to maybe 300 nanometers, but it's less than you might think given the it's a bigger distance than you might think given the resolution limit. The limitation is more stringent than we expected. Okay, so obviously if we have a very small cluster, as we have on the right, and we have a fluorophore lighting up in that small cluster, there's only a small number of fluorophores that we're prevented from imaging. In the middle, we're looking at the 1D sample, and you can see I've highlighted in red those fluorophores that we can't image. There's considerably more than in the 0D sample. And there's something like a focal adhesion, we're looking at a much bigger area again. And when we modeled this process mathematically, we saw an increase in about a factor of somewhat over 10, so about a factor of 30 between each. Sorry, I can hear some audio in the background. If anyone hasn't muted, it's probably a good idea to mute. Exciting though it is to feel like I'm really in the room with everyone. So we've got about a factor of 30 increase in the number of images required to obtain a particular resolution. And we also have different optimal activation densities to get the fastest possible acquisition. So we did all this in a purely theoretical perspective, just looking at distances between molecules and assuming all the fits would either be discarded correctly or would work correctly. In retrospect, this was a pretty optimistic assumption, and it did indeed turn out to be incorrect. But it at least gave us a clear view on what we thought was likely to happen. So we knew that we would expect an optimum activation density for each type of structure, but we also needed to be able to assess this for real data, not just for simulated data where we knew the underlying ground truth. And this is where we needed to have another technique that could assess the resolution of the data. And we used Fourier ring correlation analysis, which is still a very standard technique in the field. There has been some interesting work extending the idea of looking at correlation to use phase information from the lab of Alexandra Radonovich. And if you're interested in the issue of resolution in SMLM, I'd strongly recommend reading that paper. It's very interesting. So Fourier ring correlation analysis is an attempt to take a technique that's from EM and make it work in SMLM. And more generally, it can be used in light microscopy. So what you do in SMLM data is you take your data, which is a list of localized positions that then you can use to reconstruct an image. You split that data and reconstruct two images, and then you look at different distances in the Fourier transform. So you're looking at different length scales, which corresponds to a circle around the central point of the Fourier transform. And you look at the correlation, and you'll see that the correlation falls off as you increase the spatial frequency, that is, as you move out from the center of the Fourier transform. And you see this fall, and at some point you decide these images are decorrelated, and this is the resolution of the image. So this is a pretty neutral way of assessing resolution. I would note that, of course, splitting the information into two images is going to decrease the quality of each image, so you have to take that into account. But it is a very nice and neutral way of assessing resolution that doesn't require underlying knowledge of the sample, which was a huge challenge with methods before. I mean, the other method that had been used occasionally was to look at distances between fluorophore positions, but of course, this doesn't tell you how much information those positions are carrying. And you can use this to look at the impact of things like labeling density and localization uncertainty, which was really great. So we first decided to take this concept that there's an optimum activation density for each sample type. We would do some live cell imaging. We would start from a lower density, increase the density of the sample of the raw data until we got to where we thought we were close to the optimum activation density in both cases, and then we'd look at the difference. And we did see a factor of 30 difference in the speed required to get a given FRC resolution. So in this case, we'd started from a very low density and then gradually increased it so that we could be pretty confident that we weren't overshooting. But of course, what we wanted to do was to see that U-shape we'd seen in the original data. And so we started to crank up the, we initially just started from simulated data. We had this very high activation density and we just ran it all through FRC to reproduce our original curves. And it all went horribly wrong. So we saw these lines where we just kept getting FRC values that were better and better as the activation density increased. And that was pretty challenging because it meant that even though we had a resolution measure, when we started to get to higher activation densities, it was telling us that we had a very high resolution, even though we knew that we were getting less information in that image. And the reason for this is if we look at what happens as we go from a low density image to a high density image, we can see that the image is in fact getting sharper. So we're getting a sharper image, and that's all that FRC can actually measure. It is a measure of sharpness. And so even though there's less information in the high density images and they're less accurate, they are still sharper. And you can sort of see the pattern in here, especially in the images labeled C, where you're losing information from the central area where the information is more blurry now, and you're enhancing the brightness of the areas which are becoming sharpened. And this, I would say, is one of the central challenges to assessing localization microscopy in general, that it's very difficult to come up with a measure which will not get higher as you get something that's less accurate. And of course, that can be used on real experimental data rather than just on simulated data. So we've spent a long time thinking about this, and particularly as I did a lot of early development on high density SMLM, I spent a lot of time thinking about it because having developed some high density SMLM and spent a lot of time pondering why sometimes the results were not as good as you'd think, I came to realize that all the methods of fitting high activation density data had a disadvantage, which is that if the density of the raw data is too high, they fail, but they still give you an answer. So you will get a super res image, but this image is something the algorithm has made up. And I'm showing here a few algorithms I'm benchmarking against, but I would emphasize that we see these effects with all the algorithms we have tested. I would say they show up to a much greater extent in some algorithms than others. They kick in at a different activation density in some algorithms. Some algorithms are much more resistant to them, but it's very hard to design an algorithm which just processes single images that is not prone to this. And there's some really interesting reasons for that. If anyone wants to know about them, feel free to ask after the talk. So we have, here I'm just showing thunderstorm, single emitter, thunderstorm, multi-emitter, SOFI, and SURF, just as 4 popular examples of single emitter fitting, multi-emitter fitting, time domain, and spatial domain processing that are popular at the moment, but we have tested quite a lot of other algorithms. And the way that we developed to try and get around this effect is, however, kernel analysis, which is basically a pre-processing filtering step that you do at multiple time scales, and you follow up with standard fitting methods. So you separate your input data into events that are happening over 1 frame, 2 frames, 4 frames. And in terms of what this looks like with data, here's some input data, and you can see the different time scales showing up fluorophores at different points. And if we do this on some simulated data of parallel lines at different spacings, you can see that when we use Hawk, as we do in the last two pairs of lines in each case, we are able to get back something of the correct width. You can see there's still an advantage to using multiple emitter fits. And indeed, with other fitting methods, if you use something like SURF that's robust to multiple emitter fits, you will find that you can use it in conjunction with Hawk, and it will improve the fit. So you can use it in conjunction with whatever your favorite fitting algorithm is, and it will just stop the collapse of data into structures which are narrower than the point spread function, which are narrower than the real structure. And we tested this. So being physicists, we tested against parallel line structures, which are fortunately present in sarcomeres. And we were able to get that the information back at the same separation that we see it in EM and in single molecule imaging. We were able to get back in high density data. And we then also benchmarked against live cells. And you can see the data here. On the left, we've taken low density data. On the right, we've taken high density data. So we've got very, very different densities of data. We also did this with focal adhesions. It was a bit more challenging because, of course, you need many more fluorophores. And unfortunately, we had a slightly less well-performing fluorophore. But you can see that we are able to get back structures where we don't see these artificially sharpened structures or exaggerations in intensity differences and edge effects that you tend to get when other algorithms are used at a density which is too high for them to fit the data accurately. So I think this can be a really good approach to use in conjunction with other algorithms to ensure that you're not getting artificial sharpening. Sorry if you hear a horrific noise, that is the bin men who have decided to come around and enhance my talk with some nice high frequency sound. We're also able to use this with 3D data. Here's some data from Fang Huang on taken using biplane, where we this is an approach where we can we take information from two different Z planes at once, and then we use that to reconstruct the Z position of given fluorophores. And we're able to see, using this, using Hawk on this data, that we're able to get back information which is less prone to having data show up that's clearly very sharpened. As you can see on the left here, at one extreme, we show up some very sharpened data right at the edge of the Z range. We're able to get rid of that effect. We've also done some investigation on using astigmatism, and that also works. It is a little more complicated because astigmatism does depend in detail on the shape of the point spread function. And so you do have to do some more specific modeling that takes into account possible changes in the shape of the point spread function due to the pre-processing we're doing. And finally, what I'd like to talk about in this section on assessment is how we can use these ideas to assess data. And data assessment has been something that's been a really big issue that people have thought about for a long time, all the way back with the Fourier ring correlation paper, and there have been periodic attempts since to improve on it. One very exciting piece of work was Squirrel, which was published in 2018 by Sean Cully and Ricardo Enriquez, where they wanted to get a quantitative idea of how accurate is your reconstruction algorithm. And Squirrel had a really nice idea where you just take your super resolution image, you decrease the resolution until it's the same as a wide field image, and then you can use a reference wide field image to see if you're getting any changes in the information. So the really nice thing about Squirrel is it gives you a measure of how linear the relationship between your wide field image and your SMLM image is. And that can be very useful because SMLM is a very non-linear technique. And you can also, because of various issues to do with the way fluorophores behave and the way algorithms analyze the data, you can often get areas where you have information completely missing. And Squirrel will give you very clear warnings of this. So it can be a very good alert as to say, your algorithm has been extremely non-linear, and so you may be missing information that's very dim because the bright information has been enhanced so much, or your blinking has meant that you've completely missed this area of an image, and so it's just been cut out. So this was really good, and we decided to have a play with it. And of course, our interest is always, or has been for a while, we were definitely thinking about it then, what happens when you have high density analysis that goes wrong? And so we decided to just look at that, have some algorithms, break them, and see what Squirrel makes of it. And this brought up a really interesting issue. And I'm just going to give an example here, because we've been spending all this time thinking how do you characterize full structure when the artifacts and sharpening are highly dependent on local structure. So we thought Squirrel could solve this problem. We started out by just simulating really simple ground truth with randomly scattered fluorophores. And here I've just shown different ways of binning all that information. So that all works, but there's a really big kicker when you try and use the comparison to the wide field. And that is, if you take an image that doesn't produce sharpening like Hawk single emitter, which while it may not be the highest quality, it might not be the highest resolution, but it will not be sharpened. And then you take, say, surf, which I would emphasize, we deliberately put the activation density at a density where we knew it would break surf. And then you have the ground truth. If we look at the structure at the different length scales, you can see that in the algorithm when we know we have sharpening, we see the same structure at all length scales, whereas in the other two, we see different structure as we go to shorter length scales. And what this means is if we use the expanded wide field rather than the ground truth as our comparison, we're actually going to get a better agreement with the sharpened image. So using this method of assessment will tend to give you a higher score for the more sharpened images. And that's a real sting in the tail that we didn't expect at all. But it really inspired us to see if we could take an approach that was very complementary to Squirrel and provide the other side of the coin. Instead of asking how linear the relationship is and whether there's missing structure, could we answer the question, is there artificial sharpening? And if so, at what length scale? So we designed this to really not look at the linearity of the relationship at all. And to do this, we took our test image, which is whatever technique you want to look at. Then we used a Hawk, which is the same technique, but you do Hawk processing before. You flatten and blur the images. So you're flattening them in order to get rid of intensity information, because we want to answer the opposite question to Squirrel. They're both important questions, but you want to know if you want to know if there's artificial sharpening, this is how we're going to ask it. Whoops. We are going to say, we don't care so much about intensity information. We just care if something is there or not. So we're going to, I seem to have lost my first set of binarizations. There we go. They're showing up in a very odd way. There we go. So we binarize the image to remove that intensity information. We binarize it in 2 different ways, so we can get information about sharpening at the edges and towards the center of structures. And we then combine the image from those 2 types of processing into a confidence map. So this means that we can then identify where we're getting artificial sharpening. And if we look at different length scales, you can see if we use high density data, and here I'm taking it from the microscopy challenge data sets, you can see that if we start at very small length scales like 20 nanometers, almost everything has a warning that it's sharpened. But as you go to longer and longer length scales, you can see that gradually the information becomes more reliable. So we're able to say which length scale is the information trustworthy at. And I think this is pretty useful given how common issues with artificial sharpening are in data sets. And this has been just published, as Alex kindly said, and the tool is available for use, open source, as per usual. And we used this on different types of structures, and we were able to show that it was able to pick up structural differences. But even with all of this, we still had a sort of question that was actually partly inspired by Rita Struck, who asked, how is the information different to if you just did hawk all the time? Wouldn't that just be the most natural default state? But I think there is a way in which you can get extra information from this approach. And that is to ask, is there information present in the difference between the image analyzed with a technique and the image analyzed with Hawk? We know that Hawk does degrade the localization precision a little bit, but if you have structural differences, they may not necessarily show up as a clear structure in the Hawk single emitter image if you don't quite have the resolution. But maybe the structure will give you a difference between the images produced with and without Hawk that you can then use to identify that there is structure you will have missed. And I think this is quite useful because it's often difficult in SMLM to know when it's worth pushing to a higher resolution. You can always optimize your experiment to give you, you can always try to optimize your experiment to give you 5 nanometers more. The question is often, is it worth it? Is there something to dig out? And I think Hawk-MAN, which is what we've labeled this Hawk method of assessment of nanoscopy, is a way of also asking, is there something that I'm just missing? Should I be trying to improve my experiment? And we tested this on parallel lines. You might wonder why we always use parallel lines. It's basically because parallel lines are the worst possible thing you can use in terms of artifacts. So they're very good if you want to see if you're going to get artifacts. Now, we were certainly able to get cases where we couldn't see the structure clearly in the image, either with Hawk or with a single emitter fitting. But when we looked at the differences between the different maps, we were able to see that there were hints that there was missing structure. And we were actually able to reproduce this in a real experimental system. We looked for the Z1, Z2 protein in sarcomeres, and we're able to get an indication that there was structure below the resolution limit for both techniques that we were missing. And we were able to pick up by doing an improved experiment. And finally, of course, what we always tend to want is a resolution measure. And we have also provided that if you want to just see how your resolution is varying across your image, you can get an output where it shows you the length scale at which that particular part of the image can be considered reliable. Now, I know I've given everyone a lot of information here. we have all these different assessment techniques. When should you use these different approaches? So as a quick summary, if you think you have missing structure at scales above or at the point spread function size, or if you want to know whether your super resolved image is linearly related to your output, then Squirrel will give you the answer. If you want to know if you have artificial sharpening in your image, or whether your data is being biased by the analysis, but you are not looking for answers about linear relationships, then Hawkman will give you an answer. And if you need a number in nanometers, then you're going to want some kind of measure like Fourier ring correlation. Okay, so this is what we've worked on for quite a few years and has now been published. And I also wanted to present something which is a more recent piece of work that came out a couple of weeks ago. And that's on extracting structure from multiple images. So this is inspired by the fact that often you can image multiple instances of cell structure, and then you want to synthesize the information from these images. We've seen this with the nuclear pore complex. It's of course a very core idea of cryo-EM. But the way I've been thinking about it related to the type of image you use on the input, that in SMLM, often we have a trade-off between 2D data quality and 3D data quality. So if you can limit yourself to 2D data, you can actually improve the experiment quite a bit. You tend to do constrained model fits, but you are then constraining the possible outputs. On the other hand, if you want to do an unconstrained fit of a model, it's very slow and it can be prone to ending up in local minima. So inspired by some advances in computer vision, we decided to create a deep learning structure which allowed fitting of, which took in a whole load of images here, our input images. So we're taking in a whole load of 2D images, and we're using deep learning to fit a 3D model by fitting the rotation of the model for each of those images. And then we also perform an optimization of the structure. So we are simultaneously optimizing the rotational fit of the model to each image and the structure itself. And I think it's a neat idea because instead of simply optimizing instead of optimizing a network and then the optimized network is your output, what we, in this case, the optimization process of the network is what gives you your optimized model. And then at the end, you throw your network away and don't do anything with it. So it's the training of the network that creates this 3D model of your structure, which has been optimized without limitations. So obviously, this has some challenges. Firstly, local minima, as with almost every type of fit. And to get around that, what we did was we started out using images with very big blur, because of course, if we're rendering these images using a Gaussian from localized positions, we can image at any resolution scale that we want. So we render with very big images, very big point spread functions, and then what the algorithm will do is it will fit the general area that the molecules are in, and then gradually, as you take down the length scale of the point spread function that you're using for the reconstruction, you gradually fit finer and finer structure at the same time as you're optimizing the rotation to every single one of these images. And this is an example where I'm feeding in images at different rotations using a rotational network. You can see it's not completely getting the rotated positions. I picked one where it was struggling a little bit to give you an idea, but you can see that it is generally able to correct back and get the rotation out for a rotation in. And it's the efficiency of the deep learning network taking advantage of the smoothness in phase space to give us this relationship without an extremely long fit that would just be not practicable. So we were able to do that. And what does it look like in terms of the fit? Here we go. As you can see, we're starting out just from a uniformly random blob. And then it gradually optimizes, where it's optimizing all of the rotations. And you can see at some point it fits the structure. And we have, in this case, the Stanford bunny. So yeah, you can see we are not limiting the or constraining the fit, but it is still able for very high quality data to get very, very accurate structure out. Then, of course, we wanted to think about how will this be impacted by different processes in microscopy. We did some experiments, simulated experiments for scatter, where you can see that as the scatter gets high, it unsurprisingly starts to struggle with the structure, and also for missing fluorophores. So the fact that you only label a proportion of molecules also is a big factor that limits the accuracy. And then we were kindly provided with a data set by Suliana Manley and Christian Sieben, and we're able to reconstruct the CEP152 central protein and see the toroidal structure that they also saw in their Nature Methods paper when they did a constrained model-based fit. So I think this is a pretty exciting approach, and I'm looking forward to developing it more in the future. I'd like to thank all my group, particularly Richard Marsh, who led the Hawk and Hawkman projects, Ishan Costello, who's led a lot of the experimental work, Ben Blundell, who did all of the deep learning work, and you for listening. Thank you very much.
Thank you, Susan. That was a brilliant talk. So I think now, if anyone's got any questions, please put them into the chat, and then we'll ask people to ask them in a second. And there's a few questions there already. In the meantime, we will do the quiz quickly, which is always a experience on these.
Yes, so the link is in the chat for anyone who wants to play. And if you let me share my screen, I hope not to share my email or something weird. But indeed, just the Mentimeter, and some of you have already started to fill in the first question. So this is just to kind of get everybody excited and gathering together. Okay, just checking. All right. So the question was, in the first instance, how often do you use deconvolution? I'm not so sure what this graph actually quite means, but my answers were never when my boss asks me when my images look rubbish, and none of my images is published without deconvolution. So there are some enthusiasts who answered the last question positively, then also obviously when my images look rubbish, and then some authoritarian force when my boss asks me, some admit to this as well. And a few people said never, so hopefully Susan's talk has actually changed this and you're going to use it now more often. So let me go. I think I can.
I would 100% support those people who don't use deconvolution. I admire their dedication to pure image information.
Let me see. So I don't know if these are all the people now we have. Let me just go to the next slide. So I think this should reveal hopefully how many people are playing and who's playing. You will get some weird names associated unless you take the time to put your own name onto this. So maybe tiger or ghost or rabbit or unicorn, and a few more people signing up. Come on, guys, you can win a Foldscope, and the RMS is very kindly sending them out into the world. Whoever wins the quiz will receive one. Have you ever done some deconvolution on Foldscope images, Susan?
No, no, I think that
could be a new one.
Yeah, that could be a new one.
That just done. I did like your Hawkman image.
Oh, thank you.
That was very nice. He was
smashing. It's an interesting thing. In fact, I could have given a whole talk on deconvolution artifacts, which are.
Well, I just like the guy, so the Viking image I liked. How is deconvolution different to super resolution? So you had 3 answers here. Let's see. All right. Most people got that one right, which is great. So people have been listening, Susan. So that's nice.
Well, they were already very wise.
I think so, yeah. And then, so there's a leaderboard for this. Kirti actually loves doing this with the leaderboard. So you can see Snuggles is doing a good job in the first round, because you also need to answer quickly. So it's not just all about knowing, but it's also pressing the button quickly.
Yes, I remember. I have tended to do very slowly on all of these.
Yeah, I do them very badly too. So answer the question 2. So which techniques require image processing to obtain a super resolution image? And you have 3 answers. These ones, should be fairly easy to answer, but you should have to do it quickly. So you have 3 more. Everybody has voted. Okay, so some were very safe and said all of them, because that's often the safe one. But in this case,
so you definitely can use image processing with STED, but you don't have to get the super resolution image. But it's interesting, you know, some labs do always use deconvolution with STED, and that's quite interesting, I think.
So you don't use it with STED?
You don't have to get the resolution improvement, but the deconvolution tends to give you a much better looking signal to noise ratio, which is, I would say, why it gets used more.
I guess it's intrinsically in the software. You just kind of have it incorporated. If you use a Leica system or so.
It's just all in the depletion, but it does tend to have quite a low signal. Sorry, we didn't read this one.
Which of the following is not a method used to obtain super resolved accident information in SMLM? And I think 10 out of that.
I'm very pleased that the most popular alternative was astigmatism, because it certainly sounds like something you shouldn't put into your microscope.
Oh yeah, that's true. But the answer of spinning disc was right, and most people got it. So you can see how people are doing here now. So we lost a little bit of Snuggle, but we gained Zlatan and Boo. Mr. Micro. Who is Mr. Micro, I wonder? That'll be interesting. Okay, so number 4, I think, says.
Oh no, is it? Oh.
Did I lose one? Because it's
no, that's right.
You take an SML image of a membrane in which you see clusters. Which of these artifacts is not a possible explanation? So I had to shorten this a little bit, but I think I got it right. So maybe you want to explain a bit more, Susan.
So it's either secondary oh, sorry.
I know, so just a spherical aberration, which is not the explanation.
So that will tend to not give you clustering, but both of the others are really common. There's some very exciting work that was done on T-cell data to identify when you get these different types of artifacts.
Okay, so again, one more leaderboard, see who's catching up.
I think the next one I might have to read out because it was so
I think, yeah, that's right.
Right,
okay,
I'm on it. I have it in front of me.
So now we have, so this one is extra super difficult because the question will be said by or explained by Susan.
Right. And
so there you go.
An SMLM dataset is analyzed and the image gives a resolution of 20 nanometers using FRC. It is then pre-processed with Hawk and reanalyzed with the same algorithm, and the resulting image gives a resolution of 80 nanometers using FRC. What possible conclusion can you draw? A, the analysis algorithm does not work properly. B, the activation density of the data was too high to be fitted accurately. C, the first image has a higher resolution.
Congratulations.
Got it right.
Yay.
Good job. Good job, everybody.
That was super hard.
That was very, very hard.
That was also a first one because I just couldn't shorten it enough to make sense.
Yes.
So here we go.
Sorry about that. I should have read the lens requirements. But yeah, this was more meant to look into, maybe also emphasize that all analysis algorithms that I've tested can work properly on the right activation density, but they can also fail if the activation density is too high.
Thank you. So we have a winner, and as always, please make yourself known to either me, Alex, Alessandro, anyone you know. Put your name into the chat and your email, or Andres, or contact Georgina and Jessica, Jessica is actually a new person from the Royal Microscopic Society. We haven't introduced her, but you will receive emails from her now. So she's, the Royal Microscopic Society does a fantastic job supporting this, and we're very grateful. So, because that makes it super easy for us, and it's just a wonderful way of bringing the community together. So thank you. Thank you, everybody. So I think, are there a few, there was one question.
There's a few questions. No, there's a couple of questions. One was sent to me, so I just asked that first. I've lost it. Could Hawkman be applied to SRFF data sets?
Yeah.
That was an easy,
so,
that was a straight answer.
You can apply it with surf, you can apply it with basically absolutely any algorithm you want. So you can just do it with and without Hawk, and the Hawkman algorithm will give you warning if you're getting that artificial sharpening. I would say the only algorithm I've come across where it doesn't work great, and we are going to try and see if we can improve that, is with an algorithm called unlock, which otherwise is actually a really nicely performing algorithm, but it doesn't give data on the size of the output on the size of the fitted point spread function, and that's really useful for cleaning up the data if you get fitting to noise, which can sometimes happen after Hawk preprocessing. So I'd say that's the high performing algorithm that we haven't managed to get it to work so well with, but you can certainly test it with surf. If you have any issues at all, contact us. We've tested it with surf and it works fine. So we'd be really interested to know if you get any issues at all.
Okay, thank you. And then there's another question from Andrei Chemarov. You could ask it if they want, or I can ask. Would this all this analysis applies to mini-flux type of localization?
Okay, that is a complicated.
I think it was to the last part which you presented. It came, I think.
Yeah. So all this analysis. So certainly for localized positions, for the last thing I did where I was looking at reconstruction, you could certainly use that on min-flux data sets. But if you're thinking more about identifying artifacts, I think with min-flux, it's going to be, you will have some of the same issues, which I certainly think Hawkman could help with, but you have a more complicated scenario in that you could also have your donut-shaped beam interacting with other fluorophores and throwing off the localized position, which I think is one of the potential issues with min-flux. I mean, min-flux is of course very high performing in general, but that is a great challenge with using min-flux to do really high density reconstruction of SMLM type data. And I think that's what you would have to think about very carefully. Are you at any point activating two fluorophores when you're going to try and do the localization? So yeah, that's a fascinating question actually, because I think you would get the same problems, but you get them in the hardware, not the software. It probably require thinking about it a bit differently.
Excellent.
Do you have more questions, Alex?
I don't. There's no more questions. If anyone has any questions, you can turn on your camera and shout out at this point, or not shout out.
But otherwise, we're actually spot on time as well, because sometimes we're running over a little bit, but it's actually quite nice to be actually in time perfectly. So thank you, Susan, for the talk, and it's really great to catch up with you like this and in kind of remote world, and it's really lovely. And we have to kind of do that